Hello. We have learned about different types of splicing, the diversity of splicing machinery, that is pre-mRNA spliceosomes, group 2 cell splicing introns, and group 1 cell splicing introns. There is also another type of splicing, which is even more interesting, in the sense that it's like you have one mRNA and one pre-mRNA, and from which your one gene encodes an mRNA or pre-mRNA, and from that the splicing of exons will result in one type of mRNA. There is also splicing that can happen between two RNA molecules that are produced from two different genes, and that's something interesting. Say, for example, here we have a protein, that is, we have one RNA molecule that has produced a particular, produced from one gene, and you can also have RNA2 from a different gene. And there is a likelihood that splicing can happen, or in certain cases, splicing can happen between the RNA1 of exons of RNA1 and exons of RNA2. And that is called as transsplicing. Splicing between two different RNA molecules is called as transsplicing. Or splicing between two exons belonging to two different RNA molecules is transplicing. So the differences, you can see the mechanism remains almost same, except that the initial starting material, that is RNA1 and RNA2, are different. So there is a splicing between these two, and because they are coming from two different molecules, the formation of three-way is there, similar to what would happen in group 2 self-splicing introns. And now the splicing between exon 1 and exon 2 would occur, and then we have the splicing of spliced exons. And the intron that is left out with the three-way junction would be something like a Y structure, as opposed to what would have been the lariat structure in the other cases. I hope you can recollect that in the previous cases we have a lariat product that is formed something like this, whereas in transsplicing we have a product something like this. This is called as transsplicing. Thank you very much.